been doing in collaboration with Graham Scott at Master. So we know that for populations of organisms that invade novel habitats, ancestral phenotypic plasticity can shape the course of evolution. I just want to show you a simple example where we have a trait. Let's see that. We have a plastic trait on an ancestral population shown in black. And let's just say that a group of organisms from this population invade environment 2. And here in this plastic trait, the value is higher in environment 2 than in environment 1. Okay? So if a population of organisms buds off and invades habitat 2, if plasticity in the ancestral population is adaptive and that it brings that population towards a phenotypic optimum, we expect that selection should act to canalize that trait in that population that invades habitat 2. But of course, not all plastic traits are adaptive. Some traits are maladaptive in that they bring a population away from a phenotypic optimum. And so if plasticity is maladaptive in the ancestral population, we actually expect selection to act to attenuate that plasticity in the derived environment. So with sort of these predictions in mind, I think a fundamental question is, how does adaptive and maladaptive plasticity shape trait evolution? And then really, to get an understanding of that, we want to start to understand what genes and regulatory mechanisms underlie this evolution. And I'm going to argue that high altitude montane environments in North America are a really good model for two reasons. One, the hypoxia experience there is extreme and it's unremitting. And these sort of extreme environments have created some clear examples of local adaptation. And two, ancestrally plastic responses, so lowlander responses to hypoxia, can be both adaptive and maladaptive, and that they can either enhance oxygen transport to metabolizing tissues, or they can inhibit oxygen transport to those tissues in an environment where oxygen is really limited. And we've been using the deer mouse, Paramyscus maniculatus, as a model. They're good for a number of reasons. And we know that, well, first of all, deer mouse have the broadest elevational distribution of any North American mammal, and that highland populations from the Colorado Rockies are locally adapted to high altitude relative to lowland populations. And so I think a really interesting way to sort of test some of these predictions in deer mice is by looking at the growth of the heart under hypoxia. And that's because we know that heart size is a plastic trait. It increases along an altitudinal gradient in deer mice. It also increases with intense exercise in other vertebrates. Of course, and this hypertrophy of the heart should increase oxygen delivery to tissues that need oxygen. And that increase should be driven by an increase in the size of the left ventricle. So if you remember from your heart anatomy, this muscular region here is the left ventricle. It pumps oxygenated blood to tissues that need oxygen, like skeletal muscles, so the animal can do work. And because this hypertrophic increase in oxygen should be adaptive, should increase oxygen delivery to tissues, we expect that ancestrally plastic responses of this ventricle should be canalized in highland populations that specialize under hypoxia. We can make a completely different prediction for the right ventricle. Okay, so the right ventricle, if you remember, pumps deoxygenated blood to the lungs to pick up oxygen, right? And a key feature of lowlander plasticity is the global constriction of the lung vessels. And this is a maladaptive response. This constriction is bad. It leads to an inhibition of oxygen diffusion across the lung-blood barrier. And this can change the pressure in the right ventricle, and that can lead to right ventricle hypertrophy. So we have hypertrophy of the left ventricle that's adaptive, gets oxygen to tissues. We have hypertrophy of the right ventricle that's maladaptive. Its growth is in response to this maladaptive pulmonary vasoconstriction. And we know that in some species of highland animals, like this yak from the Tibetan Plateau, they have been able to reduce that pulmonary vasoconstrictor response. So they've been able to get around that maladaptive plasticity. So in a deer mouse, we expect the ancestral plastic response to hypoxia to be this maladaptive increase in right ventricle size, and that animals, high altitude animals that specialize in hypoxia, should show a reduction of that maladaptive plasticity. So we can test these predictions within one organ, which I think is kind of cool. And to do that, we've been taking lowland animals from the Nebraska prairie and highland animals from the Colorado Rockies, bringing them into the lab, rearing their F1 offspring at low altitude, and then acclimating those individuals for 6 to 8 weeks under conditions of either normoxia, so at an elevation equivalent to 50 meters, or hypoxia, and an elevation equivalent to 4,300 meters, which is the elevation associated with a highland site in the Rockies. So very high in the Rockies. Some of the highest peaks are at 4,300 meters. And then we took the heart out of these animals, and we measured the mass of the left and the right ventricles, and then we sequenced the transcriptomes of the left and the right ventricles separately to sort of get an idea of some of the transcriptomic signatures that might be underlying any differences in ventricle size. So I'm going to show you data on heart size in response to hypoxia. So here I have relative mass of the whole heart under normoxia and hypoxia. And what you can see is that lowland animals increase the size of their heart in response to hypoxia, and that highland animals seem to have an overall larger heart. So this is sort of fits with our predictions, except that we didn't find any statistical significance. So there's no statistical support for this, although the pattern does fit what we predict. For the left ventricle alone, we find that lowland individuals also upregulate the growth of the left ventricle in response to hypoxia. Highland individuals have overall larger left ventricles. So Highlanders have larger left ventricles, and it seems like we may have some evidence of a canalization of an adaptive response, but we don't have very good statistical support for that. For the right ventricle, we see an increase or hypertrophy in response to hypoxia in both lowlanders and highlanders. And although this increase, appears to be reduced in highlanders, we only found a statistically significant treatment effect. And this is according to a two-way analysis of covariance. I should say body mass was used as a covariate, and all these were relative to body mass. We can look at the right ventricle in a slightly different way. We can look at the size of the right ventricle relative to the left ventricle. And when we do that, what we see is that lowlanders and highlanders increase the size of the right ventricle, the relative right ventricle size, in response to hypoxia, and highlanders have overall lower right ventricles. So this suggests that at least relative to the size of their left ventricle, which is growing, highlanders may have an attenuated maladaptive response. They have a smaller right ventricle. We don't have very good statistical support for this attenuated response, though. Nevertheless, we find changes in the heart size in these animals in response to hypoxia, and we're interested in understanding what's the transcriptional basis of these differences in heart mass. We took two approaches. I'm going to tell you about those two. We took an RNA-seq approach and a differential expression approach, sort of standard. And so we mapped transcriptomic reads back to Parameciscus maniculatus genome. So we get our measure of gene expression. And for each gene, we implemented this generalized linear model to test for differential gene expression. Where we have three factors: population, whether highland is different from lowland; acclimation, whether hypoxia is different than normoxia; and then the interaction of the two. So essentially whether the response to hypoxia is dependent on the population. And we called any gene in the genome differentially expressed that a corrected p-value of less than 0.05. And so I'm going to show you those results for the left and the right ventricle separately. So for the left ventricle, what we see is, so here's the number of genes differentially expressed according to each factor in this model. So what we see is that there is an overwhelming number of genes that are differentiated between the populations. Very few that exhibit an acclimation effect, so a response to hypoxia. Very few that exhibit that interaction effect. When we look at the right ventricle, maybe not surprisingly, we see the same overwhelming signature of population differentiation. Okay, so there's clearly very strong constitutive differences in expression between the ventricles of a highland and a lowland deer mouse. In the right ventricle, we see more genes are responding to acclimation, and there are also a large subset of genes that exhibit this interaction effect. So more so than in the left ventricle. And if we look at this on a principal components analysis, so here's just a biplot of these differentially expressed genes, PC1 on the X, PC2 on the Y, what we see is that PC1 explains about 60% of the variance in gene expression. So animals, highland and lowlanders, are very clearly distinguished along PC1. And that's true for both the left and the right ventricle. And along PC2, we see that here's closed circles are normoxia, open circles are hypoxia. So normoxic and hypoxic conditions sort of are distinguished along PC2. So we see that very clearly in the left ventricle. We see the same pattern in the right ventricle in lowlanders. that normoxic individuals are clearly distinguished from hypoxic individuals here. In Highland individuals, we see that those hypoxic and normoxic treated individuals cluster very tightly together. So this suggests that in the right ventricle, lowlanders are responding to hypoxia differently than Highlanders are responding, if Highlanders are really responding to hypoxia at all. We took a second approach, which I'm going to just tell you a little bit about while I have some time. And that approach is a little bit different than differential expression analysis in that it allows us to relate variation in heart size to co-expression modules. Okay? And we did this by using a program called WGCNA, or Weighted Gene Co-Expression Network Analysis. And basically we identified a series of gene expression modules, which are just a group of highly correlated genes. So across the samples, genes whose expression varies consistently. We can identify those modules, and those modules the thinking is that they represent putative regulatory pathways. We can summarize the expression of those modules using a principal components analysis, and we give that module an expression value. It's the PC1 score. That represents the expression of that module. Okay? And then what we can do is correlate that module's expression with variation in heart size. And that's pretty cool because it allows us to associate what is a putative regulatory pathway, or a group of co-regulated genes, to variation in heart size. And when we do WGCNA, we get a whole bunch of modules. Some of them are related to the phenotypes that we're interested in, some of them are not. I'm not going to show you those results. What I just want to skip ahead and show you is what modules in the left and right ventricle are associated with size variation. And when we do that, we find this one module, which I'm just going to call Module 1, is weakly correlated with left ventricle size. And here's what that correlation looks like. We have module expression on the X and relative left ventricle mass Y. So this correlation is driven by very strong differences in module expression between Highlanders and lowlanders. And note that there's almost no variation in module expression. Look at it another way, we can see that module expression, so we're just looking at module expression on the Y now, and normoxia and hypoxia are treatments on the X. Lowlanders have consistently higher module expression than Highlanders. They're not affected by the acclimation treatment at all. So this suggests a constitutive differentiation. And a really cool thing about this is that this module, which remember is highly correlated genes, has 6,000 genes. It's about 40% of the transcriptome. So a huge proportion of the left ventricle transcriptome is differentiated between Highlanders and lowlanders. For the right ventricle, we found this one module 2 to be correlated with ventricle mass. Here's that correlation. It's a nice positive correlation. If we look at it this way, we see that lowlanders upregulate the expression of this module in hypoxia, and Highlanders have almost no expression. So that lowland plasticity, that ancestral plasticity, is absent in Highlanders, suggesting some attenuation of plasticity. So what I've shown you is that the left ventricles are larger in Highlanders, and this may reflect the canalization of ancestral adaptive plasticity, but we don't have very good statistical support. What we do know is that transcriptome-wide patterns of differentiation are very strongly constituted. The right ventricles are smaller, and they're associated with reduced signal of transcription in a single module that are tightly correlated. And I think this suggests that changes in gene regulation may underlie the attenuation of maladaptive plasticity. So I'd like to thank the NSF, the University of Montana, everyone who works in Team Deer Mouse, the Chevron Lab. If you have time and you want to see some more of the physiological aspects of this, go see Graham Scott's talk Monday, 2:45 in room 468. Thank you.